Great. So if you remember yesterday, we were talking about nucleic acids. We talked about nucleotides, nucleosides. We got into the overall structure, the double-stranded DNA structure that you've seen many times before. We also talked about RNA. One of the fundamental differences between RNA and DNA is that single-stranded versus a double-stranded structure. Now, what that means is that your single-stranded DNA or single-stranded RNA, I'm sorry, is capable of hydrogen bonding with anything and everything else that can hydrogen bond. And so what that means is RNA is very unstable. Now, with that in mind, what we're going to be talking about today is a little bit more about DNA techniques or nucleic acid related techniques. Is there anyone in here who has taken a genetics class? You probably did gel electrophoresis. Did you do the polymerase chain reaction or PCR? Okay, cool. So some of this will be a bit of a review, and I hope that I can add a little bit more to your knowledge base on that. But then we're going to talk about some kind of classical techniques that are really important for understanding basically where we are with life science research as it stands today. So nucleic acid techniques. Now, nucleic acids are very easy to work with, in part because they can be easily sequenced, duplicated, modified, and expressed. So you can take a piece of DNA and make millions upon millions of copies of it very easily. Many times it's easier to get the protein sequence or the protein sequence information from an organism's DNA rather than from its purified protein. So with that in mind, does anyone or has anyone ever heard the term central dogma of molecular biology or the central dogma of biochemistry? DNA to RNA to protein. Okay. Now, if you think about that, and I know that typically whenever that's introduced, immediately it's like, okay, well, that's good that you know that one direction, but it's actually not that simple because there's RNAs that can manipulate DNA and so on. But if you take that DNA basically going to protein as your kind of destination, then it might be like, well, I really want to know more about a protein than I do about a DNA molecule. Like if I have an enzyme that does something, I want to study that enzyme that does that thing. Well, it's easier technically, and I mean in a lab setting, to analyze or to get the sequence from genomic DNA or some sort of DNA, messenger RNA, than it is to purify a protein and cleave it, break it down into its individual amino acids and determine the sequence of that. It's far easier technically to go from DNA and extrapolate what the protein sequence is than it is to determine a protein sequence. Now, later on this semester, we'll get into kind of the techniques associated with protein sequencing because it still does have a role. But right now, is anyone familiar with PubMed? PubMed.gov. It's a great resource where you can gain access to like current literature. So if there's a disease that you're interested or curious about, go to PubMed.gov and type that disease in, and you're going to get, well, here's a paper published in January of 2024. But in addition to that, that PubMed is a great resource that houses tons and tons of genetic databases. So you can get, oh, well, what's the genome sequence for C. elegans? Well, you can determine the C. elegans sequence based on what it's been published there. So with that, you can extrapolate protein sequences. So DNA and nucleic acids are very favorable and they're very easy to work with, specifically deoxyribonucleic acids. Now, what we want to be able to do with a piece of DNA, we want to be able to cleave it into specific fragments. We want to be able to sequence those fragments, and then we want to be able to reassemble that. So is anyone familiar with the Human Genome Project? That was completed around the year 2000. Basically, here was the first draft of the human genome. Well, that's from millions of sequence reads of what is this particular sequence, then what's this, and they scaffolded it over and over again to basically construct Well, of all of the different chromosomes and sister chromatids within an organism or within a human, here's the sequence and here's how we can put it all back together. And that's what we want to do with proteins. We also want to do it with nucleic acids. We're going to focus on it with nucleic acids and how we do that. Now, one way, bless you, that we do that is with a type of enzyme known as a restriction enzyme. These will sometimes be referred to as restriction endonucleases. Now, what these do is these restriction enzymes specifically cleave double-stranded DNA. And you might say, well, double-stranded, isn't that kind of implied? It is kind of implied, but this is a point to notice. Well, restriction enzymes don't do anything to single-stranded DNA. You're not going to see single-stranded DNA too often, but if you did, a restriction enzyme is not going to do anything to them. These restriction enzymes were originally purified primarily from or isolated from bacteria. Now, there are three different types of them. Type 1, type 2, and type 3. The differences between these types are what are the sequences, the primary structure of a piece of DNA that they recognize. A type 1 restriction enzyme is going to require ATP, and it's non-specific. Whenever I say non-specific, what I mean is that this piece of, or this, let's say we had a piece of DNA that looked like this. And I'm just going to write one strand, 5' to 3', but I want you to understand that there is that reverse complement strand. So if we had a piece of DNA that was A, T, G, C, A, and you've got the opposite strand that aligns with that. And then we've got another piece of DNA that is A, C, G, A, T, A. This isn't a trick question or anything like that, but are those two sequences that I've written, are they different from one another? They are. They have a different primary structure. A type 1 restriction enzyme has a non-specific cleavage pattern. So what it does is it sees that and it says, aha, double-stranded DNA. That's what I'd like to cut. And it's going to break that. So it's going to say, oh, okay, I'll break there, and then I'll break there, and then I'll break there, and then I'll break it there. It just kind of almost arbitrarily cuts a double-stranded piece of DNA. Does everyone follow that? Okay. Now I want to contrast that with something like a type 2 restriction enzyme. This does not require ATP, but it has a specific cleavage sequence. Now, what that means is that, let's say that we have, well, I'm going to go into those in just an example. What that means is that a type 2 restriction enzyme is going to take a double-stranded piece of DNA. It's going to jump into those major and minor grooves, and it's going to read the base pairs. It's going to say, okay, I'm looking for I'm looking for a specific sequence. I'm looking for an ATC. And whenever I see that ATC, I'm going to cut after that C. Does that make sense? If it says I'm looking for ATC and it sees ATG, it's not going to do anything. It's going to keep processing until it sees ATC. Does that make sense? Okay, cool. Then our third type is going to require ATP. It also has a specific cleavage pattern. So types 2 and type 3, they are specific. They know what they are looking for. The difference between them is type 2 does not require ATP in order to hydrolyze that bond, and type 3s do. Now, in the next slide, we're going to look at some examples. This is a long list of examples. I have no expectation that you memorize these or learn these. What I do want you to understand, though, is this right here, this enzyme name. Now, I'm going to highlight a couple. Maybe the most common restriction enzyme is a restriction enzyme known as EcoR1. I've highlighted it. It's written as ECORI. Okay. Now, I want to break this name down for y'all for just a moment. If it's R1, now this is a restriction enzyme. If I were to say eco R1, and then I were to ask you, what do you think the R stands for? As it is a restriction enzyme. What do you think R possibly stands for? Restriction. Okay, that's a good place to jump. Now, next up, the next character, 1. Do you think that this could possibly be the 10,000th restriction enzyme found from a particular bacteria? Or maybe it's the second, or maybe it's the first? Let's go ahead and presume that R1 stands for restriction enzyme 1. Is everyone good with that? Now, if we were to look down at, say, something like eco-RV, thinking about your Roman numerals, if the R again stands for restriction, which enzyme do you think that this one is found from a particular bacteria? It's not the first. Thinking of our Roman numerals, what's the V stand for as a Roman numeral? 5, the 5th. So would everyone kind of see, oh yeah, I can see how that could be the 5th one. Okay, now let's chat. Perfect. So now if we are to go back and let's go ahead and look at one other component of this name. Where did I say that these were purified from? Bacteria. Okay. Now, is there anyone in here who's taken a microbiology class? Okay. What's a bacteria that is kind of your most simple lab bacteria? Does anyone like to say E, the E part of it? I love to say it. Escherichia. Oh man, it sounds so fun. It's also the word that I struggled with for a very long time, and I was like, escargot. So E. coli. Okay. What do you think ECO stands for? E. coli. So this particular bacteria, when you see ECOR1, this was the first restriction enzyme discovered in E. coli. Does that make sense? Now, all of these other notes, ECOR5, it's the 5th restriction enzyme extracted from E. coli. All of the other names, all of these, PVU, I think that's a Pseudomonas vulgaris, not a microbiologist, but that 3-letter or 4-letter or sometimes 5-letter scheme that's italicized is basically saying this is the organism that it's purified from. So there's a method to that madness. Now that's one part that I want you to be familiar with whenever you see it. Am I going to ask you questions about what bacteria is this from? If I do, I'd probably give you the E. coli one. But I want you, whenever you see that, to be like, aha, I know I can make a presumption based on bacteria that I know where this probably came from. Does that make sense? Now let's follow that up. Great. With the next column of this, we have restriction sequence. Is this another? So one new message. Yeah, I saw that. Recognition sequence. Okay. Now these are all specific sequences. These sequences are highlighted. So based on that, is this a type 1, type 2, or are these type 1, type 2, or type 3 restriction enzymes? So I'm going to bounce back. Type 1s require ATP, non-specific cleavage. Type 2, no ATP, specific cleavage. Type 3 requires ATP, specific cleavage. So if you see that sequence, are any of these type 1s? No, they're not. Because they have a specific sequence that these enzymes that they are going to seek out and then they're going to cut. Does that make sense? Now, do we know or can we say whether they're type 2 or type 3? Not based on what's on this table. But we do know that these are the specific sequences. Now, what I want to do is I want to focus in on EcoR1, GAATTC. Now, GAATTC is a specific sequence that we're reading this from the 5' to 3' direction because we always read DNA in the 5' to 3' direction. Now, this is a double-stranded piece of DNA. So I'm going to go ahead and draw this. G, A, T, T, C. So there's one of our strands from our 5' to our 3' end. Now we have a reverse complement. That is going to be G. Actually, you know what? I think I'm going to change color. I'm going to go with a blue just to give it contrast as a different strand. That G is going to have how many hydrogen bonds between G and C? 3. Okay. Then what complements T? A. So we got an A here. And we got an A here. What do we have? What complements A? T. Okay, so we got a T, T, and then finally we got a C. Does everyone see that? So this end is our 5' end, just as this end is our 5' end. 5' to 3'. These are reverse complements of one another. Okay, now let's imagine that this piece of DNA is somewhere within an organism's genome. Well, EcoR1. Think about, I always visualize an enzyme as like this blob. This blob is going to jump on a piece of DNA, double-stranded DNA, and it's going to be looking at the sequence. And it's going to be going in this direction. It's going to be just kind of looking in those major and minor grooves. And what this blob, this EcoR1 is going to do is it's going to have a restriction sequence that it recognizes. It's going to say, I'm looking for a G. And you know what? It's gonna look at both of these strands, right? It's gonna look here and it's gonna be like, well, I've got a C up here, but I've got, oh, that's a G. Yeah, good, good. What is on the 3' end of that G? Well, the 3' end of that G is an A. Does everyone follow me there? Now, I'm bringing it up with this lower strand because you got to remember, these are reverse complementary strands. One strand, the one that we wrote first was GAATTC. The other one, we're reading from right to left as opposed to left to right. So reading from right to left, is it GAATTC? It absolutely is. So what this enzyme is going to do is it's going to see that G, and then it's going to see AATTC. And what it is going to do is it's going to break the bond between that G and that A. Does everyone follow that? What this thing is also going to do, as it's kind of scanning along the length of this piece of DNA, it's going to see another place where we've got a A on the 3' end of a G, right up here. And it's going to cut there. Okay. Now, my question that I love to ask is right here and right here. When it breaks a bond, what type of bond is broken between the G and the A? Now, you got to think back to the double-stranded piece of DNA. Is this really a bond directly between a guanine and an adenine? No, it's a bond between those sugars. What is the bond that connects our sugars in a double-stranded piece of DNA?
Is
One
it
of your classmates? Say that again.
I said polymer chains, but I feel like that might be wrong.
It's a phosphodiester. So it's a phosphodiester bond that's between our two sugars. So this bond right here, phosphodiester, which one of your classmates did the thing that I know that I've done many times in my life, and that is try to say it loud enough, but without moving your lips. So then if you're right, you can be like, I did it. And then if you're wrong, you can be like, who said that? That was this phosphodiester. I saw it. And yes, you're absolutely right. So it's a phosphodiester bond here and here. So restriction enzymes, they directly break phosphodiester bonds. Now, these two bonds that are broken, are they directly across from one another? No, they're not. They're staggered from one another. Now, with that in mind, that gives way to some of the different products that are produced from DNA cleavage. Here we have another visual of, on the left-hand side, of the action of EcoR1. It's going to cut on the 3' end of a G and on the 5' end of our A. So we see that here. We see that same thing here. This is a tricky thing to do because you're thinking about these two strands, they are reverse complements of one another. So it definitely takes a minute to like reorient yourself when you're reading that other strand. So we have this phosphodiester bond that's broken and this phosphodiester bond that's broken. Now, between all of our nitrogenous bases, we see those 3 dots. What do those dots indicate? What type of bonds? What are the types of bonds that hold the 2 individual strands together as one complete thing? Hydrogen bonds. So it's those dots correspond to hydrogen bonds. So now the question that I love to ask is, what types of bonds are directly broken by a restriction enzyme? Phosphodiester bonds. Now, when you take something like this cleavage pattern that's produced by EcoR1, you've broken these 2 bonds. It's almost like you have a bridge where you've broken like it's almost like a suspension bridge, like the Mackinac Bridge in Michigan, or the Golden Gate Bridge in San Francisco. There's those big wires that if you were to break those, what's going to happen to that bridge? It's going to collapse. Now, here, what we've done is we've broken those huge cables that hold that bridge up. We've broken those. Those cables are our phosphodiester linkages. Have we directly gone to basically knock down the supports underneath that bridge? No, we haven't. The hydrogen bonds are the supports underneath the bridge. But when we break the cables that hold that suspension bridge up, we've destabilized it enough that the hydrogen bonds are not adequate to keep this one molecule intact. Does everyone follow that? So we've destabilized it, or we've broken it by destabilizing it. So the question that I love to ask, and that I love to think about this, is when we break the disulfide or the phosphodiester bonds, those are the only bonds that our restriction enzyme is going to directly be breaking. Are hydrogen bonds ultimately broken? Yes, they can potentially be, when the 2 cleavage sites are not directly across from one another. As is indicated on the left-hand side here, we have 2 phosphodiester bonds that are broken, but then what we get are hydrogen bonds that are broken kind of indirectly. Does everyone follow that? So it's common that recognition sites in double-stranded pieces of DNA usually have a 2-fold axis of symmetry. These are palindromes, so GAATTC. Well, reading on one strand and then the opposite strand is the palindrome of that. Now, enzymes are going to recognize sequences that are 4, 6, or more bases in selecting for a cleavage site. Now, type 2 enzymes, EcoR1 and EcoR5, are examples of type 2 restriction endonucleases, and they have different recognition sites. What make these different from one another is, one, the specific sequence that they identify. EcoR1 is GAATTC, and EcoR5 is GATATC. Now, EcoR5, it is not going to cut after a G. It instead is going to look for GAT and cut on the 3' end of that T. Does everyone follow that? Now, what types of bonds are or what type of bond is EcoR5 going to directly break? It too is going to break phosphodiester bonds. Now, when we look at EcoR1, can everyone, this is an example that I'd like you to play along with me with. Take your left hand and your right hand. Now, my middle finger is longer than my index finger. What I always like to do is do this. So I see some like overlapping regions where the tips of my middle fingers are touching one another. So I've got one hand where my palm is facing me, and then one where the top of my hand is facing me. So these are the patterns, or this is the pattern that I'm going to get whenever I have something like EcoR1. So if I look at my left hand, I have my left index finger and my right middle finger. Those are tip to tip. Does everyone see that? Now, one of my EcoR1 cut sites is going to be at the end of my index finger and at the beginning of my middle finger. So I'm cutting here between the tips of my fingers, and that's where I'm breaking my phosphodiester bonds. So as soon as I break that, now all of a sudden, the force that's holding my hands together, it's no longer a phosphodiester bond. It's only the hydrogen bonds holding the tips of my middle fingers together. Is everyone with me? Okay. Now that's not strong enough. Think about the tips of my middle fingers are holding my arms together. Well, my arms are stronger than those 3 bonds that are holding my middle fingers together, in which case that's going to break. Now I'm going to have a product that looks like this. Does everyone see that? Where, yeah, one strand of my DNA is my middle finger, one strand is my index finger. This is referred to as a sticky end with what is known as a 5' overhang, because the 5' end of one of my pieces of DNA, my middle finger, is longer than that of my index finger. Now, this is another thing where it's like, I always have to take a pause and I'm like, okay, I follow that it's a 5' end, but what about on my other strand? Well, it's still a 5' overhang because I'm still reading 5' to 3' ends. So here's, there you go. It's perfect, right? Okay, so we have broken phosphodiester bonds first. And then because we broke those super strong covalent phosphodiester bonds, now all of a sudden those hydrogen bonds aren't enough to hold the molecule together. EcoR1 has a cut pattern where we have that 5' overhang. That is an example of sticky ends that are produced. Now the other outcome from a piece of double-stranded piece of DNA being cleaved is where you have something like EcoR5. When you look at EcoR5, we're cutting 2 phosphodiester bonds that are directly across from one another. So rather than it being like my index and middle fingers, it's almost like the pieces of DNA are my fists when I put them together like this. One phosphodiester bond is right here at the top of my fist, and the other is at the bottom of my fist. When I break those, no hydrogen bonds are broken. There are no 5' overhangs. Does everyone follow that? So these are referred to as blunt ends, which I always hear that and I'm like, okay, that kind of makes sense because my fist is effectively a blunt object. It doesn't have a point to it or anything like that. So when you think of restriction enzymes, there's a couple of things that I want you to know. One thing that I want you to know is they come from bacteria. 2, there's a little bit of a naming scheme to it. The number corresponds to basically which one it was that was discovered from a particular organism. The recognition sequence, there are 4 nucleotides or 6 nucleotides or 4 base pairs or 6 base pairs long. They can be even longer than that, as we see with the one that I kind of scribbled through, BGL1 or Bagel1. That has a sequence that's a little bit less obvious than others. Now, I bring all this up because these cut pieces of DNA, these cut pieces of double-stranded DNA, these effectively were or are kind of a defense mechanism. A bacteria has this defense mechanism. EcoR1, if it cuts GAATTC, and this is E. coli existing in the universe, it has an enzyme that cuts GAATTC. My question for you is, do you think that that specific sequence, GAATTC, is found anywhere in the E. coli genome? And before you answer that, let me ask you this. Why would E. coli want to cut its own genome? And let me rephrase it with facial expressions and tone. Why would E. coli want to cut its own genome? And then I'll rephrase it again. Would E. coli want to cut its own genome? No, I don't want to cut my own genome. Now, let me ask you this. Do you think that E. coli's like biggest enemy, do you think that GAATTC is found in its biggest enemy's genome? I'm seeing some slow nods of yes, yes, because if that organism, if this other E. coli were to try to, if this other bacteria were trying to basically take over any space that E. coli was in, well, E. coli is going to be like, to hell with you, here comes some EcoR1, and it's going to cut your genome up, which is a nice way of saying, I'm going to burn your house down. Bacterial way of saying, I'm going to burn your house down. Does that make sense? So these are the, yes?
Sorry, I was gonna ask, so basically what you're saying is like the restriction enzymes are meant to break like the DNA from like competing bacteria.
Perfect, absolutely.
Okay, cool.
Okay, so blunt ends and sticky ends. Sticky ends have 5' overhangs, blunt ends have blunted ends. Okay, a technique that maybe you've seen before is gel electrophoresis. Gel electrophoresis is a technique used for separating pieces of DNA. It's also used for separating pieces of protein. Here, one thing that if you've done gel electrophoresis that's maybe a little bit different than what you've seen before is this gel is a vertical gel as opposed to a horizontal gel. The same principle exists. We separate DNA and we can separate pieces of DNA based on their molecular weight or based on their size due to DNA having an overall charge that is negative. Since DNA has an overall negative charge due to its sugar phosphate backbone, the larger the piece of DNA, the more negative charges it has. The smaller piece of DNA, the fewer number of negative charges it has. Larger molecules will stay at the top because they're moving through this complex matrix. Smaller molecules will migrate towards that positive anode a little bit easier. Now, restriction fragments, I'm not going to go into this too deep just because I want you to know that restriction fragments and restriction mapping is an old technique that basically you could use to sequence a gene. If you ever look up any if you go to graduate school and you look up any sort of dissertations in biochemistry, molecular biology, microbiology, from like the late 70s, early 80s, you'll see that someone earned a PhD seemingly just by sequencing a gene. Now, if you've worked in a lab or done anything like that, now it takes like 24 hours to sequence a gene. You have your piece of DNA, you send it off, boom, you get your results back the next day. Well, in the 70s and 80s, it was a 5-year-long process. And you would do something called restriction mapping, because what you would do is you'd have your piece of DNA and you'd cut it with one enzyme. And you say, okay, well, when I cut it with this enzyme, here are the products that I got in my DNA gel. Now, when I cut it with a different enzyme, here are the products that I got. And what you're doing is effectively mapping. You're mapping using these different enzymes. So if you want to find out 1,200 base pairs, the sequence of 1,200 base pairs, you're doing it 4 base pairs at a time, 6 base pairs at a time. And so you're basically cutting and saying, okay, well, because this is cut in the middle by EcoR1, that means I know GAATTC is in the direct center of this gene. So as you can imagine, that's a very meticulous and laborious process. I like using both those words as much as possible. So what you would do is you would cut with one enzyme and you would get two pieces, just as an example. We're cutting one piece of DNA with two enzymes, or sorry, we're cutting one piece of DNA with HIND3 and it gives us two products. We'd be able to say, okay, well, that makes sense because our piece of DNA was 4 kb. It was cut here, here's the products. Then we would cut with another enzyme. And here's the result that we would get. And then we would cut with both of those and we kind of piece it back together. We know that there's a BAMH1 site here, there's a HIND3 site here, there's a BAMH1 site here. This is a process known as restriction mapping. Yeah, this is ultimately, in quick summary, this is a lot like a logic puzzle, if anyone's ever done one from like middle school, where it's like Joe wears red shoes, Sally wears blue shoes, Dave is taller than Sally, who's the oldest person in the group? You're finding these connections between different people, and then you're putting an order to it. So this is the same sort of thing. You cut a piece of DNA with this enzyme, with both enzymes, you piece all that information together. You're scaffolding information and you're putting it all together. That's restriction mapping. Restriction enzymes are useful for construction and cloning of recombinant DNA, the mapping of DNA and DNA fragments, assuming random distribution and equimolar proportions of our 4 different nucleotides. A particular tetranucleotide sequence occurs every 256 bases. Now, this is a math question that you see on my exams. So if you are, basically this math is, if you have a piece of DNA that's 2 nucleotides long, what are the chances in this specific site that you would get an A? This is a randomly generated piece of DNA. We're randomly generated. What are the chances that you get an A? Huh? 25%. And then after that, what are the chances that you get a T in the second location? Before you answer that, does whatever you get in the second location have any, or is it influenced at all by what's in the first location? No, it's totally random. So it's 1 out of 4. So it's a quarter that you get that A, and then it's another quarter that you get that T. So the chances that you see AT in a randomly generated piece of DNA, 1 out of 16. Does everyone follow that? So if you randomly generated a piece of DNA that was 16 base pairs long, there's a pretty high probability that you'd get an A followed by a T. Is it 100%? It's pretty darn close to it. Does everyone follow that? So then, if you're talking about a restriction site that is GAATTC, what are the chances that you find a G? 1 out of 4. A, 1 out of 4, 1 out of 4, 1 out of 4, 1 out of 4, 1 out of 4. So you're talking about 1 out of 4 to the 6th power, which is, you would find that once every 4,096 base pairs. Because it's just a quarter times 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 a quarter. Does everyone follow that? Okay. Okay, now next up is a technique that restriction fragment linked polymorphisms. Restriction analysis of chromosomes is extremely useful in determining inherited genetic defects and obtaining other genetic information. Human chromosomes differ in sequence every 200 to 500 base pairs, creating or eliminating restriction sites. So what that means is you can take a piece of DNA from me, you can take a piece of DNA from one of my kids or someone else, and try to cut it with EcoR1. And me and my kids might have the exact same cut pattern, but me and somebody else will not have that same cut pattern. Does everyone follow that? Okay. Treatment with restriction enzymes yield fragments with different lengths, or what is known as a restriction fragment length polymorphism, which is one of my favorite acronyms in science, and that is a RIFLIP. Mutational change resulted in the loss of that restriction site. So that would be the difference between me and some random person. And so that's one of the ways that we can do paternity testing or relationship testing. And basically what you do is you take pieces of DNA from multiple people, you cut it with the same enzyme, and you run it on a DNA gel. Now, this right here is a technique that uses that exact same thing, but uses a radio labeled probe. If I remember correctly, this is a southern blot. I always get, there's like there's multiple different blots. There's a western blot, southern blot, northern blot, a northwestern blot, a southwestern blot. And they all have to do with the different things that you're trying to detect. If I remember correctly, when you're detecting DNA, it's a southern blot. But that might be it. I'm not going to hold you out to that. The next technique, we've got two more techniques that I want to go over, and then I'm going to go ahead and stop for the day. PCR, the polymerase chain reaction. PCR is cycle that is repeated over and over and over again. This is a technique that allows you to take a double-stranded piece of DNA and go from one copy of that double-stranded DNA to millions of copies of that double-stranded piece of DNA. From a single piece, you get multiple others. So the first step in this first cycle, this right here, what you're going to do is you're going to be separating the double-stranded piece of DNA by heating it up. And whenever you heat it up, those two strands of DNA, what are they doing? They're separating into two individual strands. Okay, so what type of bond is being broken between those two individual strands? I'll give you a hint, it starts with an H and ends with a hydrogen. Hydrogen bonds. So we're using heat to separate those pieces of DNA. Hydrogen bonds are being broken. Are phosphodiester bonds being broken? Nope, they're untouched. So hydrogen bonds, we're going to separate into two individual strands. Then what's going to happen is we are going to get these things right here, which are indicated right there. These little blocks of DNA that are single-stranded that perfectly anneal to our two single strands. Those are known as oligonucleotides. They have a complementary sequence to each of your two strands. So essentially what you do is you take a double-stranded piece of DNA, you separate into two single-stranded pieces of DNA. Then you have these oligonucleotides that jump onto those single-stranded pieces of DNA. Now, this thing right here that I'm highlighting, your eyes are not deceiving you. That's a single strand for most of it, but there's also a region of it right here that is double-stranded, where your oligonucleotide has bound to it. So you have the molecule that's totally highlighted right here is partially single-stranded and partially double-stranded. Does everyone follow that? Now that region that is double-stranded, and let me clear all my markings up. This region right here, this is my region that's double-stranded. What that is going to do is that's going to attract an enzyme known as DNA polymerase. DNA polymerase says, I love it whenever I see a piece of DNA that's partially double-stranded and partially single-stranded. Whenever I see that, jump onto it. And then what I'm going to be able to do is I'm going to bring in DNTPs. That lowercase d, what does that indicate?
Deoxy.
Deoxy. At what position do I not have an oxygen? 2' the 2' position. That's good. We work together and we put that all together. It's the 2' position on my sugar. So that lowercase d indicates that I am synthesizing DNA. Does everyone follow that? So this DNA polymerase right here is going to see that double-stranded region. It's going to bring in DNTPs, and it's going to extend this double-stranded piece of DNA. Does everyone follow that? Okay. So basically what's happening is I heat this up and break a bunch of hydrogen bonds. Then I'm going to cool it down so that clear all this so that these regions can be formed. I'm going to cool it down so that those regions can be formed. And then my polymerase is going to go to work. So the heating takes place at approximately 98 degrees Celsius. The cooling, where the annealing takes place, where my new oligonucleotide primers bind, is going to be at approximately 55 degrees Celsius. And then my DNA polymerase is going to go to work at approximately 72 degrees Celsius. So I'm going to cycle between these temperatures. That's cycle number one. Then what I'm going to do is I'm going to kind of rinse and repeat. I'm going to separate my newly synthesized pieces of DNA, breaking a bunch of hydrogen bonds. Then I'm going to have more oligonucleotides bind, and then I'm going to extend. And I'm going to continue doing this approximately 30 to 35 times. Nope. 30 to 35 times. Now, some more details on that. These steps are known as denaturation, hybridization, polymerization. Denaturation is where we break the hydrogen bonds. Hybridization and denaturation takes place at 98 degrees. You might see it sometimes as 95 degrees. We're talking something close to the boiling point of water. Hybridization takes place at a cooler temperature, approximately 55 degrees Celsius. That's where we're rehybridizing, where we're making hydrogen bonds between our oligonucleotide primers and our single-stranded pieces of DNA. And last but not least is our polymerization, where our DNA polymerase synthesizes the new strand of DNA. And that's taking place at about 72 degrees Celsius. Our requirements for this, we have two flanking primers. These are our oligonucleotide primers that bind to our single-stranded pieces of DNA. We have a heat-stable DNA polymerase. Heat-stable, what do you think that means? It can exist, it can still function if it gets all the way up to 98 degrees Celsius. In addition to that, it can be heated up, cooled down, heated up, cooled down, heated up, cooled down, and go through that 30 some odd times. If you think about ice cream, if you take ice cream and you let it melt and you put it back in the freezer, is that going to be good anymore? No, it gets weird. Unlike a DNA polymerase or this specific DNA polymerase, it still functions. So it's heat stable. It can endure those temperature changes. We also need the 4 DNTPs. So we need DATP, DGTP, DCTP, and DTTP. And then last but not least, we need a specific divalent cation. That divalent cation is magnesium, Mg2+. Mg2+, that divalent cation is important because of the fact that magnesium, what it's going to do is it's going to stabilize the alpha and the beta phosphates on an ATP molecule or a GTP or a CTP or a DTP or TTP molecule. PCR as a technique that's invaluable for forensics. We have DNA fingerprinting as a method. It's also been employed in restriction fragment length polymorphism analysis because you can take that small sample of DNA, amplify it, and then cut it, and then see the relationship between people's medicinal detection of infectious bacteria and viruses. Go get your PCR test because otherwise you can't come to the concert, sort of thing. But obviously it's not just exclusive to COVID or anything like that. Okay, how's everyone feel about PCR? Okay, do we need ATP or DATP for PCR? We're making DNA, so we need the ATP. Okay, good? Okay. Now this next technique, DNA sequencing. This is a process known as the chain terminator procedure. This is a technique that is going to use a different DNA polymerase. It uses a fragment of that DNA polymerase known as the Klenau fragment. This can use a single strand of DNA and a DNA primer, nucleoside triphosphates, DNTPs, just like PCR, but here's where there's a little bit of a wrinkle to it. We're also going to use a small amount of 2', 3' dideoxynucleoside triphosphates. So DDNTPs. So if you think about ATP versus DATP, ATP has a hydroxide group on the 2' and 3' carbons of ribose. DATP or DNTPs do not have a hydroxide group on the 2' of ribose, but it has a hydroxide group on the 3' of ribose. DDNTPs or a 2' 3' dideoxytriphosphate lacks a hydroxide group on the 2' and the 3' of ribose. Okay. And that's effectively what it's going to look like. So if you remember for DNA, that's our 2' carbon where we lack that hydroxide group, but we keep it here. Well, when we're talking about a DDNTP, we lack that OH group. Okay. Now, with that in mind, let's think about a piece of DNA. Whenever you're synthesizing a piece of DNA, you need to have a hydroxide group there so that you can elongate it. You can use that phosphorylated group to basically make a phosphodiester linkage. If you don't have an oxygen here, you cannot make a phosphodiester linkage. So that's where a piece of DNA synthesis stops. Does that make sense? So at this point in time, the biggest distinction between PCR and DNA sequencing is DNA sequencing also involves a dideoxynucleotide triphosphate. Other than that, they're calling for largely the same things. This dideoxynucleotide triphosphate terminates a reaction. Now, here's our example of a template piece of DNA and a primer strand. So we are adding new nucleotides to this primer strand. As long as we see that hydroxide group right there, that's going to react with this phosphate to make our phosphodiester linkage. Does everyone follow that? Because that oxygen has some lone pair or has lone pairs of electrons. So we can make that phosphodiester linkage. Now, if instead of an OH, we have an H, do we have those lone pair of electrons so that we can make that phosphodiester linkage? We don't. Okay, so that's the importance of the 2'3" or 2' 3' dideoxynucleoside triphosphate, the DDNTP. Now, what this is going to give us is we're going to get a sequencing gel, which is something that looks like this figure on the bottom. Now, what we will do is we will do a total of 4 different reactions. There's one. Here's the other. Here's the other. And here's the other. Okay. So we are doing 4 separate reactions simultaneously. Now, for the purposes of this, I'm going to go ahead and just perfect. So I have a reaction that I'm going to refer to as my A reaction. My A reaction has all 4 deoxynucleotide triphosphates, which are indicated right here. Okay. My A reaction also has a small amount of DDATP. My enzyme is going to look at my template, and it's going to be reading it. And whenever it sees a T, what is my enzyme going to do? My enzyme is going to see the nucleotide or the nitrogenous base of thymine, and it's going to say, okay, I know what matches up with a T. What matches up with a T? Hey, perfect. Okay, and so think about this as the enzyme has this bag, and this bag, it's got 4 different bags. It's got one bag that's DCTP, one that's DGTP, one that's DTTP, and then one that's DATP and DDATP. Okay, so whenever it sees a T, is it going to go to the DTTT bit? That bag? No, it's not. It's going to say, I need to get an A there. So it's going to reach its hand into that bag, that bag that has DATP and DDATP. If it pulls out a DATP, it's going to put it on there. And since it's a DATP, we're going to be able to continue making the piece of DNA after that. Does everyone follow? If in the event that it puts its hand in that A bag and pulls out a DD ATP, well, that DD ATP is going to be a terminator. And we're going to get a product that looks like this. Because what's happening is, let's go ahead and imagine that we're making this piece of DNA. GG are a part of the primer. The DNA polymerase sees the G and it says, okay, I'm going to get a C. Done. Then it sees the next G, pulls out a C. Done. Then it sees a T. It goes into its bag and it says, all right, well, I'm going to pull out an A. And I happen to pull out a DDATP. And I'm going to put that DDATP there. That DDATP is not going to allow me to produce any more of this bond because it doesn't have that lone pair of electrons, because it doesn't have a hydroxide group. It just has a hydrogen. It's a dideoxy nucleotide triphosphate. It looks like this right here. Does that make sense? So what that means is that we are going to get a product that's a total of 5 nucleotides long, 5 base pairs long, that first one. Does everyone see that? Okay. Now, we've got thousands of copies of this going on, thousands of copies of the enzyme, thousands of copies of the template. So this product is going to accumulate, but simultaneously, there's going to be an instance where our Klenow fragment, the enzyme that we're using, pulled out a DATP and it put an A in that location because it's random chance, because it has both DATP and DDATP. Does everyone follow that? So it happened to pull out a DATP and put it in opposite that T. And then we were able to continue on making the remainder of this product until it encountered another T where it pulled out a DDATP to complement that T. Does everyone follow that? So when we look at a gel like this at the bottom with our A reaction where we see 2 bands, which is what we see right here, what does that tell us about how many T's are in our template? If we see 2 bands, that means there were 2 products produced, right? So how many T's are in our template? 2. I really like it whenever I ask a question like that I'm intending to be like, this is a good, easy question. And then maybe this is just me reading into it too much, but I get a response of 2. I'm like, yes, exactly. We didn't need the attitude, but yes, you're absolutely right. It's 2. Okay, does everyone follow that part? Okay, so that reaction's taking place. In addition to this, we've got other reactions that are taking place. We've got our C reaction. And from our C reaction, we've got all 4 of our deoxynucleotide triphosphates and we've got our dideoxy CTP. So wherever there's a G, we can either pull out a DCTP or a DDCTP. If we see 3 bands, how many G's are there in our template? 3. Our G reaction. There's 2 C's. Our DDTTP. Well, there happen to be 3 A's because we use that. There are 3 products from our T reaction. Now, this is all valuable because we can take all of this information and treat it like everyone's favorite Canadian TV superstar, hip hop superstar Drake, because we're going to start at the bottom. And we're going to go there. I think that's the song title. We have all of these different products that are generated of different lengths. And what we're going to do is we're going to say, which is the shortest fragment that we have? The shortest fragment was from our C reaction. So that means that we have a C. in the first position of our product. Our second shortest fragment is also from our C reaction. Our third shortest is from our A reaction. Fourth shortest is from our T, C, G, T, T, G, A. And ultimately, this is going to give us a sequence that complements our template. So all of this is valuable because we can say, oh, okay, well, from our 5' to our 3' end, we have C, C, A, T, C, and so on. And then you can extract from this, well, what is the sequence of my template? Well, if this is a complement to my template, I take the reverse of it, and that gives me the template's sequence. So this is a technique, and this is a way that years ago, this was how DNA or pieces of DNA were sequenced. Now, however, we use fluorophores and fluorescent labels where we do basically the same thing, but now you get a readout like this shown on the lower right-hand corner, where you can get DNA sequence very, very quickly and not really have to worry about a DNA gel like this. You can piece together, okay, well, here's the exact sequence based on here are the products that were generated. Okay, good. I'm gonna leave out the recombinant DNA part just because if you take a molecular biology class, that's what you would cover in there. In the meantime, I'm going to stop my share. I'm going to stop my recording.